Okay, now we're going to shift our focus from the mechanisms driving water movement to the actual pathway that water is moving through as it makes its way through a plant. And the vast majority of the pathway is made up of cells that are a part of the vascular system. So the vascular system is just the generic name for that tissue that is transporting water. And there's a couple specific types of cells that we're going to talk about. So here's just initial picture. I'll tell you about them and we'll come back and talk about them in more detail. The two pictures at the top are called vessels or vessel elements, and these are found in angiosperms. The diagram on the bottom, these are called tracheids and they're in gymnosperms. The important parts that I want you to recognize right now is that this right here that I'm drawing around right now is a single cell, a single vessel element. And it's generally fairly open-ended on each end. So they're like these pieces of pipes all stacked together. Tracheids, on the other hand, you can see the ends of the tracheids actually taper and close. On the wall of tracheids are these pits that water can move through. So to go up a plant, a water molecule has to go up one tracheid, pass through the wall, and go up through another tracheid. And it can never pass through the open end of that cell because the end is not open-ended. Vessel elements also have these pits. There you can see these little circles on the bottom here. Those are pits. So water can also move out of, one vessel element and go into another one, but it can also go straight through most of these vessel elements really more easily. So moving through these pits is a little bit slower than just going straight through the open end. All right. So those are an introduction to vessel elements and tracheids. Let's talk about water moving through these cells. So again, these cells are kind of like stacked on top of each other and they're beside each other and they're overlapping. So water moves up through some of them, maybe multiple ones. It might move to the next one over and move on up. But the way we kind of quantify how fast water can move through these cells and through a plant is by, we can calculate the hydraulic conductance, all right? So here I've got the equation we use for conductance. And this equation has been around for a really, really long time, and it's just the same equation mechanical engineers use to calculate conductance of a pipe or a hose or anything else that's moving fluid in an outside of a plant. And so we're just using that same equation for plants. So I'll just walk you through what's involved in this equation. It's fairly easy to calculate, and we'll do an example calculation in just a second. So you've got pi in here because we're talking about circles. R is the radius of a vessel or of a tracheid. So whatever pipe you're talking about, that's going to be the radius of that pipe, to the power of 4. That's in the numerator. In the denominator, you just have this constant 8 times L, which is the length of the pipe. And for us, we're going to do the height of the plant. All right, so the length of those pipes is however tall the plant is. And then P is the pressure gradient. That's the water potential gradient from the bottom of the plant to the top of the plant. So usually what we'll do is we'll use the soil water potential and the leaf water potential. The difference between those is the water potential gradient. All right, and then this equation here is really for one pipe. Then we have to multiply it by the number of pipes a plant has, and that's multiplied by this n. So that is what goes into this equation. So let's just do an example calculation so we can see kind of what the impact of this radius is on conductance. So I'm going to draw again. Here I've given you some measurements for two different plants, all right? So L of both plants is 1. So we're pretending, these are just made up numbers now to give us kind of realistic numbers. So we're talking about a 1 meter tall plant, so we're talking like a shrub. The water potential gradient for both of these plants is 1.5 megapascals. The radius for the one on the left is 0.5 millimeters. The one on the right is 1 millimeter. The number of pipes in both of them is 100. So everything's the same except for the size of the pipes. And that'll give us an idea of how important the size is. So if we do this calculation, for this one we're going to have, see if I can write legibly, not very, times 0. Boy, this looks like my kids when they were in, I don't know, kindergarten. I'll get better. That's all over 8 times, I have a lot more respect for Khan Academy now. L is the height of the plant, so that's just 1 meter, and the water potential gradient is 1.5. All right, and then this one is going to be 3.14 times 1 to the power of 4 times 8 times 1 times 1.5, and then I forgot to multiply it by the number. They both have 100. And if you do those calculations, this one ends up about a conductance of 1.63, and that's basically, ooh, that was a horrible circle. I'm having fun drawing. I hope you're not miserable. 1.63, and that's really how many molecules of water are moving through these pipes per second. And then on this one, it's 26.16. All right, so it shouldn't be a surprise, all else equal, the plant that has the larger radius cells or pipes has higher conductance. But what I want you to do or think about is if we take the ratio of these two, so if we take 26.16 divided by 1.63, the difference is 16 times. What that means is if you double the size of a pipe, you don't double the ability to move water, you increase it by a factor of 16. So a relatively small change in the diameter or the radius of a pipe has a huge impact on how much water it can move. So if you are a plant where there's lots of water and you need to get that water up to your leaves fast so that you can have wide open stomates as long as possible and maximize photosynthesis, you're going to want to have large pipes to move that. All right? So that's how big of an impact the radius has on it. I have this slide again, but we don't really need to talk about it again. Let me just show you a figure, and we're going to talk about this and some trade-offs here with these pipes in just a second. So the figure on the left shows K sub leaf by A max. All right, K sub leaf is the hydraulic conductance of the leaf. So we just calculated conductance. This graph shows the conductance for an entire leaf versus its photosynthetic rate. And what you can see is that plants that photosynthesize fast, high up on the y-axis, have to have really fast conductance. They have to be able to move water really fast through their leaves. And that's because they have large vessels. That's how they do that. All right? And we'll get to why they use large vessels it'll be more on the assignment, and we'll come back and talk about it next week. So K-leaf is the hydraulic conductance of a leaf. The larger the value of K-leaf, the more easily and quickly that water can move through the plant. And so how does this affect the growth rate? And so if you can move lots of water, you can keep your stomates open for a long period of time and wide open so you can maximize your growth. But remember, when I said that we treat this water in the plant as a continuous column of water. So it's kind of this fragile entity, single entity almost, within the plant. And if something disrupts that column of water, it's going to disrupt the ability of water to get from the soil up to the leaves, and that will affect photosynthesis. All right? So what can happen in plants is if the tension on the water column gets too great, the water column can actually break. So let's just, now I'm really hesitating to draw because it's so hard to draw on this thing, but I'm going to do it anyway. So let's say for an example, oops, let me get the draw on. Let's say we have a vessel, right? We've got a single vessel, all right? And let's say, all right, it's got some water in it, all right? Now, there is tension on this water column, all right? I'm going to draw some arrows representing the tension. And that's because, let's say the leaves are at minus 1.5 megapascals, and the soil's at minus 0.5 megapascals, all right? Neither of these are under positive pressure, right? They're both under negative pressure. Both of them have some pull in their direction. The soil, if the soil was a positive megapascal, it'd be pushing water up the plant. But because it's negative, it's also kind of like holding on to its water. So there's this tension on the water column between the soil and the leaves. If that tension gets large enough, what happens is an air bubble, you won't have to remember all of this, but air gets pulled in one of those pits, and when the air gets pulled in, it creates a water bubble, and it breaks this water column. All right, so I'm just going to draw a breaking. So the air gets in and breaks that water column. So now that water column is no longer continuous, and so water is not going to be moving up towards the leaf in that one pipe. All right, so this happens a lot in plants. And if 1 or 2 or even 5 pipes breaks in a plant, or the water column breaks, not a huge deal. I mean, it's going to affect the water movement, but not a lot. It's when you get lots and lots of these pipes with the water column breaking that we end up with a problem, okay? What happens, I'm going to kind of go back. What happens after this column breaks? What happens is our vessel, let's do a different color. I don't know if this is going to be a big enough difference. Oh, all right, that looks like air. Now this is air filled. That doesn't work, does it? Now this vessel is full of air, not transporting water. Sorry. And this is referred to as an embolism. I want to get this on here. Don't share these slides with anybody because I don't want them to see how bad my penmanship is. Okay, so this is what happens if the tension on the water column gets too big or too great. So you can imagine during a drought, the tension on the water column is going to be very large, and so we're going to potentially get some of these cavitation events leading to lots of embolisms. So it turns out that larger vessel elements, so these ones that have a larger radius, they're a bit leakier to air. And so what that means is that air gets in easier, and so there needs to be less tension on the water column to pull air in and cause an embolism in plants with large vessels. So here's just some data showing this trade-off. So on the x-axis, I have, this is the water potential that causes a 50% reduction in conductance due to cavitation. Take-home message is, as you move from the left to the right, you get less embolism formation. All right, so and each one of these dots is a different plant. So this is a plant that does not easily get embolisms, so meaning small or narrow pipes. These plants down here get embolisms very easily. They have large pipes. But you can see the plants with large pipes can conduct water really quickly. And then these plants with small pipes and are resistant to embolisms, they have low conductance, they can move less water and less easily. So what that means, there's a trade-off. There's a trade-off between the ability to transport water, K-leaf, and the ability to tolerate drought, that embolism formation. So that means having large pipes is really good for moving lots of water, but it's not so good for moving water when soils are dry. So that's one trade-off associated with this pathway for water movement. Again, here this figure shows these are plants growing in lots of different places around the globe. You can see this paper, if you want to see where these data are taken from, the paper there will describe these sites. But what we have is the mean annual precip of a site on the x-axis. So these are dry places, these are wet places. And then we have that P50, or the water potential that causes 50% loss in conductance. Again, the more negative this value, the more resistant those plants are to embolism formation. All right? And so for drought, maintaining growth under drought, that's a good thing. So what you see here, we can look at this log transformed data here that shows it nicely. Plants growing in wet areas, they are not able to resist embolism formation because they have large pipes. Plants growing in dry places are able to resist embolism formation because they have these smaller pipes. All right, so that's the trade-off with drought. Turns out there's also a trade-off with freezing tolerance, or freezing. I'll show you the graph and then I'll do another amazing drawing. Okay, so here is the relationship between, this is mean conduit diameter. So this is like the radius of your vessel element. And this is the percent loss of conductivity. So 0 means that all of the conduits, or all of the vessels, are full of water and are moving water. 100% loss of conductivity means all of those vessels are embolized and that none of them can transport water. And this is, so the open symbols are plants that were just grown under happy conditions and measured. And you can see in general, these are all relatively low. They're not, a lot of them aren't 0, but they're relatively low. Then these closed symbols represent plants that were frozen and then their conductance or conductivity was measured. And what you can see is that the plants that have the larger diameter or larger I tried to say vessel and radius at the same time. Larger radius vessels, those ones experienced lots of embolisms and so lost all of their ability to conduct water. So if you're a plant growing in the far north of North America, you don't want to have large pipes because they're going to most likely embolize when it freezes, and then they can't transport water. And if you can't transport water, you can't have your stomates open, you can't get CO2, you can't photosynthesize. All right? So that's kind of what this pattern is showing, the relationship between vessel size and sensitivity to freezing embolism. Let me just show you what happens during freeze events. So if I'm going to draw another vessel here. Okay, this is a vessel, and when this vessel freezes, it will get an air bubble in it. Okay, so it's just what happens when water freezes inside these vessels. And remember, these air bubbles can be a really bad thing, right? Oops. Try to do a race. Okay, so these air bubbles are a bad thing inside the cell. I'm trying to get some water in there. Sorry, this is taking a while. All right, there's our water. All right, but then there's an air bubble that forms inside. And this is always going to happen. The plant can't do anything about it. What's important is when this water thaws. When this water thaws, a lot of times this air bubble just goes back in, dissolves into the water, and like the water can continue transporting. It doesn't form an embolism. There are two things that can lead to an embolism after freezing. One is bubble size, And the second is water potential. Okay? The larger the bubble, the harder it is to get back into water. The more negative the water potential, the less likely the air is going to go back into water or dissolve back into water. Okay? So if you have, let's draw two vessels. When these freeze, the one on the right has a much larger bubble. And so that's going to be harder for that bubble to go back into solution when it thaws. And so let's say both of these were at minus 1.5 megapascal. I'm not going to draw the units. That means this one's probably going to cause an embolism at minus 1.5, but this one's really small and it won't form an embolism at 1.5. But if the water potential gets low enough, this one might also form an embolism. So what this means is plants that use large vessels to transport water, they're more sensitive to embolisms due to freezing. And so they generally don't grow in very cold places. And they certainly aren't functioning during cold times of year. But plants that have small vessels, they're really good at tolerating those cold temperatures. And so you'll generally find them dominating colder areas. Here's just another, so an example of the trade-off between freezing tolerance and growth. So on the y-axis, we have the stem freezing tolerance, and on the x-axis, this is the annual growth rate. You can see plants that are good at tolerating freezing, so plants that are good at tolerating freezing have low growth rates, but plants that have high growth rates are really kind of sensitive to freezing temperatures because of the size of vessels they tend to use. So there's this direct trade-off between the vessel size and drought and freezing. Okay, I want to continue on, even though this is getting long. I was trying to keep my videos to 10 to 15 minutes. I've failed, sorry. I want to talk about the vessels and the tracheas a little bid more, and then we'll move on to nutrients. So I mentioned that there's two types of water transporting cells. There are tracheids and there are vessels or vessel elements. So tracheids, again, are only found in gymnosperms. They're good at resisting freezing and drought. And that's because they're really narrow and they're closed at both ends. And because there's only one type of, okay, this is just an example of the wood. I'm getting ahead of myself. All right, now let's summarize the vessel elements. So again, here's a picture of a vessel element where you've got these open ends on the vessel, and they tend to be larger radius than the tracheids. So they're larger and they're open-ended, so they move water really quickly, but they're not good at tolerating freezing temperatures, and they're not good at tolerating drought. The other thing that's important about vessel elements is because they're so big, they're not as structurally sound as tracheids. And so in angiosperms, they have these vessel elements, but in order for trees to grow tall, they also have to use another cell called a fiber. And a fiber is this tiny little cell, or it's really narrow cell. It doesn't transport water very well, but it provides lots of structure. And it moves almost no water at all, so we don't even account for it. But it provides lots of structure. So the important part is that angiosperms use two types of cells in their wood, and one to transport water, one for support. But in gymnosperms, tracheids serve both purposes. Why I'm bringing this up is because you can then tell the difference between angiosperm wood and gymnosperm wood by the variability in sizes of the cells. The two on the top, the two pictures on the top represent angiosperm wood, and this picture on the bottom represents gymnosperm wood. Let's start with the gymnosperm. So between these two arrows here, that's 1 year of growth. So this represents an annual growth ring. And you can see for almost this whole year, the cells are pretty much the same size. I mean, if you go in there, you can see some smaller ones and some big ones, but they're relatively similar in size. And even at the end of the year here, they're getting a bit smaller, but you can still tell an individual tracheid apart from another one. Up here, on the other hand, are the angiosperms. And so again, between these two arrows, that's 1 year of growth. You can see these large open areas here. Those are cells. That's the inside of vessel elements. That's the white part. Then this part that looks orangish and you can't really distinguish an individual cell, those are the fibers. So you've got vessel elements are really big and easy to see, and fibers you can't tell the difference. So there's this drastic difference in cell sizes in angiosperms, and you can just see it by looking at the wood. This is another example of an angiosperm. So here's the two growth rings, or the in between these two arrows is the annual growth ring. And you can see the larger, in this case, they're dark in the center. That's the inside of the vessel elements. And then the other part of stuff you can't really distinguish, those are the fibers. And that's because of the difference in the types of cells they're actually using. This is just another slide summarizing the gymnosperms. This slide summarizes the angiosperms. I said all of this information already, but it's here on the slides for you. And then lastly, I'm just going to point out, you might have noticed the difference between the two pictures I showed you of the angiosperm wood. There's two different types, or there's two different ways that trees kind of distribute their vessel elements. One type is called ring porous. And so these cells here are produced in the spring. And so these trees that grow this way, they put all of their big vessel elements, they grow them all early in the spring, and they use those to transport water all summer long, or all during the entire growing season. This is ring porous. Then after they grow these cells, they produce a bunch of fibers. So all of their large vessels are right at the early part of the ring, or in the spring. Diffuse porous is the opposite, where they distribute their vessel elements all throughout the annual growth ring. So they're continually building new vessels throughout the year. You don't see this big pulse of them in the spring. They're distributed throughout the entire year. And there's some functional differences between those two types of wood and the trees that produce them. And if you're interested in that, I can guide you to some of that literature, because it's really cool, some of the functional significances of these differences in structure. Okay, that's all for water. I'm going to take a short break. I'll make a new video for nutrients.